Thanks, Charlie, and thanks everybody who's been doing a great deal of super work to get this to start and keep Club EvMed going as well as it is. It's really been a highlight of this COVID environment, and we've had some really fabulous speakers. And so when the invitation to introduce today's speaker came out among our group, I jumped at this one. It's my great pleasure to introduce to you Dr. Christine Stabell Benn from the University of Southern Denmark. She's an MD and PhD who has done just pioneering work in the field around the world, particularly in Guinea-Bissau, one of the poorest countries in the world, to ask questions about the role of vaccines in training our immune system in ways that we may not expect. And just based on today's attendance, it's clear this is a question of broad interest. We also learned briefly just before we started about the Institute for Advanced Study that she has had a hand in founding at the University of Southern Denmark, which sounds like a really exciting environment, which may indeed have positions down the road in this area. I won't say anything more because I know we want to hear from Dr. Benn. Please take it away.
Thank you so much for the kind introduction, and thank you for inviting me to speak here. I'm thrilled to be in your club. I attended one of the sessions before today, and I really loved it. And I look forward to talking with all of you who have a bit of a different background than people I usually talk with. And that also means I might have gotten the wrong idea about what you would immediately understand or what would be difficult to understand in my presentation. So I really want to extend Charles' invitation to raise questions during the talk, and just say, do unmute and ask your question along the road if I'm saying something which doesn't make sense or you need to have clarified. But let me get started and share just a proportion of all the research we've done in the interest of time, but just to give you an appetizer and hopefully get a good discussion. And I'll start with a brief disclaimer. You have to do that when you talk about vaccines these days. So apart from being a medical doctor and an epidemiologist, as I was introduced, I'm also the mother of 2 kids who received all the vaccines in the Danish vaccine program, just to make sure that there's no doubt about that. And I also want to disclaim that I've spent part of the last 27 years in Guinea-Bissau, in a small West African country, studying the overall health effects of vaccines. And as such, I became a co-developer of the concept of non-specific effects of vaccines, and that obviously makes me biased in that regard. But apart from that, I don't have any conflicts of interests. According to the current paradigm for vaccines, a vaccine is a biological preparation that improves immunity to a particular disease, and that understanding guides how we evaluate and monitor vaccine, because we do that usually based on studies which assess the effect of vaccines against the vaccine disease, or sometimes even just on biomarkers for the vaccine disease. So unknown to most people, all the currently used vaccines have never been evaluated for their effect on overall health. We've simply been so sure that vaccines just prevented the vaccine disease and nothing else, so it hasn't seemed necessary to conduct studies before they were introduced, looking at the effect of these vaccines on overall mortality and overall morbidity. That effect has been based on extrapolations. So if a measles vaccine, for instance, was 90% efficacious and measles was 70% of all death, 10% of all death, then it has been estimated that, well, introduction of measles vaccine in a naive population would reduce overall mortality by 9%. But these assumptions have never been tested in real life. These are extrapolations made behind the desk based on the knowledge of the specific effect against the vaccine disease. So our research conducted in Guinea-Bissau at our field station has led us to challenge that paradigm for vaccines. We have a field station there in in this small country with 1.9 million inhabitants, situated in West Africa, and we follow with a health and demographic surveillance system 100,000 people in the capital Bissau and a similar number of people in randomly selected rural villages. And we collect all kind of demographic information on pregnancies, on deliveries, and on mortality, mobility, and we also collect health data on vaccinations, infections, hospitalizations, etc. And that permitted us to raise the question that hadn't been addressed before, namely, what is the effect of vaccinations on overall mortality and overall morbidity using all the data that we had collected? And we very quickly found out that there was something missing in this equation, because what we saw was that we couldn't at all predict the effect of a vaccine on overall health based on the knowledge of its protective effect against the vaccine disease and the knowledge about the prevalence of that vaccine disease in the community. So, for instance, for measles vaccine, we saw that when measles vaccine was introduced, it reduced all-cause mortality by more than 50%, by 70%, which was much more than anticipated based on the effect against measles infection. And for other vaccines, we saw actually the opposite, that even though a vaccine had a very strong protective effect against deadly diseases, it wasn't followed by the anticipated reduction in all-cause mortality and morbidity. So that led us to propose the hypothesis that vaccines, in addition to their protective effect against the vaccine disease, also have what we call non-specific effects on other diseases. And the effect on overall health is the sum of these two effects. The non-specific effects were seen in a context where the risk of dying was mainly due to infectious diseases, so it was clearly a non-specific effect which was targeted towards the immune system and its ability to handle other infections. Before I tell you what we found in more detail, I just want to explain that there are two main categories of vaccines. There are the live vaccines and the non-live vaccines. And the live vaccines are mostly old vaccines. These are the vaccines that contain the pathogen in a weakened, attenuated form. So it creates a very mild disease in your body. Usually it's so mild that you don't feel any symptoms when you get the live vaccines. You might have a slight fever or so, but you have this micro infection, this mild infection in your body, and that gives a broad and long-lasting immunity. In contrast, we have the non-live vaccines, and they contain the dead pathogen or inactivated pathogen or parts of the pathogen, products of the pathogen, or gene material from the pathogen. And these non-live vaccines are not as good at creating immunity. They're not as good at stimulating the immune system, which cleverly enough doesn't really interpret these non-live vaccines as a threat. And this is why the non-live vaccines often have to be given together with a helper substance, an adjuvant. And these are the vaccines you remember because you have to go back to the doctor several times to get them, because you need your primer and your booster and more boosters. And some vaccines you even need to have, 10 every 10 years and so. So the non-live vaccines, you could think, are not as good because they didn't create such a good immunity, or at least it took longer to create the immunity. But they have some advantages. They are more stable than the live vaccines, which have to be kept in a cold chain, etc. And also, they can never create the real disease in the host, even if you're immunocompromised. Whereas in rare situations, the live vaccines can create the real disease in immunocompromised people. So nowadays, most of the newly developed vaccines are non-live vaccines. These vaccines create good specific immunity. You might need some more doses, but in the end, you'll end up with good specific immunity. But it turns out when we look at their non-specific effects that they are very, very different. Because the pattern we have observed after studying now 10 vaccines for their non-specific effects is, and this is 40 years of research really, which is summarized in this slide. The pattern we have observed is that the live vaccines, and there we looked at measles vaccine, BCG vaccine, smallpox vaccine, and oral polio vaccine, and also measles vaccine in the form of MMR vaccine. All these live vaccines have beneficial non-specific effects. So we saw the same pattern, that their introduction and their use versus no use is associated with much larger reductions in all-cause mortality and morbidity than anticipated based on the specific effects. But worryingly, and this is why I have to state up front that I vaccinate my own kids, we have this signal from low-income countries with high mortality from infectious diseases that the non-live vaccines, and there we looked at DTP vaccine against diphtheria, tetanus, pertussis, hepatitis B vaccine, pentavalent vaccine, inactivated polio vaccine, H1N1 influenza, and RTSS, and new malaria vaccine. These vaccines are not associated with strong reductions in mortality as anticipated. They are associated with the opposite, actually, and intriguingly, this is seen for females. So for all these 6 vaccines, we have seen that females who receive them have higher overall mortality than the females who haven't received them, and then the boys who have received them, this is in spite of the specific disease protection. I'm not going to elaborate more on that right now, but happy to take any questions in the discussions regarding these last findings. But I'll just show you some of the nice findings on the live vaccines. Here, the first study I alluded to that my mentor Peter Arby did when he introduced measles vaccine in Guinea-Bissau in 1979. I was 11 years at that time, so not really around. But what Peter saw was really what kickstarted this research agenda, namely that vaccinated kids with measles vaccine had 70% lower mortality than unvaccinated kids, and measles is easy to diagnose. It was easy to exclude that the non-vaccinated children had died from measles. So this was pointed towards this beneficial non-specific effects against mortality from other infections of measles vaccine. And numerous observational studies have been done confirming these findings also from other parts of the world. And I'll just jump directly to the randomized trial we were able to do. It's actually quite difficult to get to do randomized trials with vaccines that are already introduced, because you cannot get ethics permission to postpone or not to give certain vaccines which are already part of the program. So you have to be a bit ingenious, thinking up study designs which allows for the possibility to assess vaccinated versus unvaccinated kids. But in the case of measles vaccine, we did this trial where we had all children receiving measles vaccine at 9 months of age, which is the current recommendation in Guinea-Bissau, but we randomized them to receive a measles vaccine, an early additional measles vaccine at 4 months of age, or no such measles vaccine, permitting us to assess in an unbiased fashion the effect of receiving 1 versus 0 measles vaccine, and later on 2 versus 1 measles vaccine up to the age of 36 months. And here are the results of this randomized trial, basically confirming what we saw in observational studies, that receiving this early measles vaccine was associated with 30% reduction in all-cause mortality up to 36 months of age. And if we censored for measles, there were very few measles deaths, but censoring for these measles deaths really revealed that the majority of this mortality reduction was due to non-measles, to reduction in non-measles deaths. We also looked at BCG vaccine in a randomized trial, and here again we could take advantage of the fact that low-weight neonates are not vaccinated with BCG at birth, which is otherwise policy. They only get it postponed around 6 weeks of age with their first DTP vaccine, and this created an opportunity to randomize these low-weight babies to BCG at birth and the usual OPV, or just OPV, which would be the usual treatment, and then follow them for mortality in the neonatal period to look for the effect of this BCG vaccine against all-cause mortality. And we did 3 trials with 3 different mortality settings or context in the control group here, as you can see. And in all 3 trials, we saw that there were substantial reductions in neonatal mortality during the first month of life in the group that received BCG at birth versus the group that was randomized to just do as usual and receive it later on. So in a combined analysis, BCG vaccine was associated with more than a third reduction in neonatal mortality. And this was clearly not due to tuberculosis, which doesn't kill babies in the first month of life. It takes long to develop. So we could show with verbal autopsies that this was due to reductions in the risk of dying from pneumonia and from septicemia. So pointing towards and supporting beneficial non-specific effects of BCG vaccine. And again, in this interest of time, just showing very briefly the randomized trials, but we have numerous observational studies which support this observation. And in 2014, WHO conducted a review of the non-specific effects of BCG and measles vaccine, and also of the DTP vaccine. And the results were published in 2016 and supported and really corroborate what I've shown you here, that receipt of the two live vaccines, BCG and measles vaccine, seemed to reduce overall mortality by much more than anticipated. And you can see the meta estimates here of the many observational studies and randomized trials included in the review, pointing towards reductions of almost a halving of child mortality associated with these two live vaccines. But you'll also see that completely in contrast here, we have the effect of diphtheria, tetanus, pertussis vaccine. The effect was just not significant in its own right, but it's highly statistically significantly different from the effect of BCG and measles vaccine. And it is a worrying and concerning finding, because we cannot think of any explanation why, or any bias that could explain that this should be due to a bias and not due to a worrying negative effect of DTP vaccine. You may ask then, why would a vaccine, or how could a vaccine affect the immune system so broadly to actually create such substantial effects on overall mortality? And there we were really greatly helped by the research conducted by a great colleague at Radboud University, and these are some of his slides. It's Mihaly Tia, who has been the father of the concept of trained immunity. His research has challenged the traditional view of the immune system, where we have thought about the immune system as the innate immune system and the adaptive immune system, and really thought about the adaptive immune system as the place that builds immunological memory with its B cell and T cells. So traditionally, the innate immune system has been seen as a kind of as some foot soldiers that you put out in front of yourself, in front of, while you prepare the true army, the adaptive immune system. And these foot soldiers are pretty stupid. They enter the battle and they exit the battle with the same knowledge. They haven't learned anything in the battle. They don't have the memory of what's going on, and it won't affect their subsequent meeting with another pathogen. This has been the traditional view, in spite of the fact actually that the adaptive immune system is quite unique. It's only 5% of all living species that have an adaptive immune system, and plant and insect people have known for decades that the innate immune system is actually pretty well functioning, it's pretty smart, and it has a memory. So Mihaly Tia took this knowledge back into human research and into the laboratory and into human experimental studies, and was able to develop a new concept in human immunology, namely the concept of trained immunity, and the fact that this traditional view is really not sufficient to explain what happens in the immune system, because the innate immune system indeed also in humans turn out to have a memory. It learns something from a first encounter with a pathogen, which shapes its properties and also results in a subsequently very different response when you meet a second infection, whether it's with the same pathogen or an unrelated pathogen. So the reason why we got in touch with Mihaly Tia was that he picked up on our epidemiological findings and started looking at BCG vaccine. And he did a study in Dutch volunteers where he gave them BCG and drew blood just before and 2 weeks and 3 months after. BCG vaccine is the vaccine against tuberculosis. And he saw, as you could anticipate, that there was an increase in the innate immune cell cytokine responses towards Mycobacterium tuberculosis, which is the pathogen, target pathogen. But he also saw that there were increases in responses to Staph aureus and Candida albicans, so completely different pathogens. And he was able to show very neatly that all this was related to epigenetic modifications in the monocytes, where the monocytes genes for pro-inflammatory cytokines were turned on by the BCG vaccine. So this, of course, is not indicating that this might have an actual physiological effect, but we were able to do a really nice little experiment together, a proof of principle experiment, where we took Dutch volunteers and gave them BCG vaccine or no BCG vaccine. And then we challenged them 4 weeks later with a yellow fever vaccine as a kind of challenge infection. Yellow fever vaccine is a quite harsh vaccine, so it gives viremia. You can measure the virus in the blood. 5 days after you get the vaccine. And what we could show was that there was much less viremia in the blood of the volunteers that had received BCG prior to yellow fever vaccine. And we could show that this was related to these epigenetic modifications in the monocytes induced by BCG and resulting not least in the production of more IL-1 beta, a cytokine which again correlate negatively with the virus load. So this really documents that a vaccine with a bacterium which should not affect anything else according to the current paradigm for vaccines than the actual target disease, that this vaccine modulated the subsequent response to a completely unrelated pathogen, in this case a virus, subsequently. And this, not least this study actually, and this experiment, I think has led to a great interest in whether BCG vaccine might affect the course of COVID-19, might prevent or COVID-19, or at least prevent severe COVID-19. So as researchers into non-specific effects of vaccines, we have of course been worried by the pandemic, but on the other hand also extremely thrilled that this seemed to have really ignited a huge interest in non-specific effects of the live vaccines and whether they may have a role against not only COVID-19, but also in the toolbox against future pandemics. So right now, we and others are looking into BCG vaccine against COVID in randomized trials. We are doing trials with oral polio vaccine against COVID. And I know in the US, there's a huge trial ongoing with 30,000 participants, I think, looking at the effect of MMR vaccine against COVID. So this will be super exciting to follow what happens with these trials. And I don't know whether COVID is a strange virus, whether it's actually affected by these non-specific effects of vaccines, but no matter the outcome, I think we're going to learn so much from these trials that are ongoing now, also about the effect of vaccines in adults, because so far we have mainly studied it in children. Yes, I'll just have a few last words about maternal priming, because this is something we are so interested in for the time being. These are new findings of ours, of the importance of maternal findings. And again, just gazing back at what is known in the insect and plant world can teach us a lot about what happens in humans, because I read this article in 1999 when it came out in Nature. It was a commentary on a study done in plants where they saw that if a plant is attacked by a larvae, then the plant's defense response includes the production of bitter tasting toxic compounds. But also this study showed very intriguingly that if the offspring of the radish will also have an increased propensity to produce these bitter tasting toxic compounds. So the title was lovely. I thought it was, if you attack the mother, you will have a hard time when you then subsequently try to fight the daughter. She will be much better prepared for that fight. And I think that was maybe what I had in mind when I got the idea to study whether the maternal vaccination with these live vaccines could somehow affect how the offspring would respond to the same vaccine. And indeed, this seems to be the case. So here I show you the results for BCG vaccine, or just so far 2 of them. We have more in pipeline. But what these 2 studies show, I'll start with this one in Denmark, where we looked at in a randomized trial at the effect of BCG vaccine versus no BCG vaccine for children and the risk of subsequent infectious disease hospitalizations. What we saw was that there was no effect of BCG vaccination to the offspring in mothers who hadn't been BCG vaccinated, but in mothers who had been BCG vaccinated, we saw a strong beneficial effect if the offspring received BCG versus just saline. And in Guinea-Bissau, a similar finding, slightly older children, where we looked at their BCG scar status. We know BCG scar is a strong predictor for lower mortality, but it turned out when we could stratify by maternal vaccination status that this was only the case if the mother herself had been primed. And we have exactly the same findings for measles vaccine. We have now 5 studies just submitted for publication where we can show uniformly that children who are vaccinated in the presence of maternal antibodies against measles, but not maternal antibodies against other pathogens, that this specific combination of maternal measles antibody in the baby and the baby receiving measles vaccine is associated with very strong survival benefits. So I'll wrap up by just because you're the ones you are asking a few thoughts or throwing out a few thoughts I've had about what this could all mean in an evolutionary context. One thing we have talked about often is that the live vaccines are, many of them contain the pathogens that we co-develop with, and it really begs the question whether we somehow also have some survival benefits of meeting these old pathogens, and also the implications that we maybe need to discuss whether we should continue to use BCG vaccine in countries with no tuberculosis just for its immune training effects. And a very important question in relation to eradication. We stopped smallpox. The whole idea with vaccines is really, to vaccinate until you eradicate the pathogen, and then you can stop the vaccination and harvest all the benefits. We did that for smallpox vaccine without anybody ever questioning when we stopped smallpox vaccine whether it could have any consequences. And it turns out we have now several studies of historical cohorts that actually smallpox vaccine was associated with beneficial non-specific effects. And stopping smallpox vaccine may have actually been a really bad idea for our overall health. And we have 2 diseases coming up for eradications, not least measles, but also polio, and the question that this begs is whether we should continue vaccinating also if we eradicate these pathogens. And yeah, the other thing is I'm just intrigued by the finding of the maternal priming effects and the idea that it would be particularly beneficial to meet a pathogen that your mother has met. So perhaps we are more top-tuned than we actually thought to meet the same pathogenic, but perhaps in general, the same environment as our mothers grew up in. And I'm wondering whether we could exploit that, for instance, by vaccinating adolescent girls with the vaccines that we anticipate to give their babies also. So I've shown you some results which I, together with many other results, think challenge the current paradigm, because we have so much evidence now coming from epidemiological studies and also from immunological studies that vaccines are not only a biological preparation to improve immunity to a particular disease, they also have effects on other pathogens, other infections. And that means that this current system we have in place for evaluating and monitoring vaccines is simply not enough. It's not enough to focus on vaccines and assess them by their effect on the vaccine disease. We need to take the broader perspective, even though it might be complicated sometimes, and look at their effect on overall health. We cannot just base our assumptions on extrapolations. So in a recent review, we asked the question whether it's time to change the paradigm. And in this review, there's much more information than I've been able to provide here. Anyway, this I think was enough for me. I just want to acknowledge briefly all the many people who have contributed to the research that we have done over the years, just a fraction of them here, and Mehanetia in Nijmegen, at least for a wonderful collaboration around non-specific effects of vaccines, and not least all these mothers and children in Guinea-Bissau who turn up every morning with their kids, trusting us to do our best to make sure that they get the right treatment and the best probability of surviving. And this is what I will continue to strive to do. And with this, thank you very much for your attention, and I hope there are many questions.
Well, wow. So there's certainly many questions and a tremendous enthusiasm in the chat. I want to get right to it, but we have the good fortune of having a discussant here from our own Center for Evolutionary Biology and Medicine at Pitt, Doug Reed, who's in our Center for Vaccine Research here, who has been working on vaccine development for multiple respiratory pathogens here, bacterial and viral, and has been thinking about questions like yours. Doug, if you want to just if I can invite you to unmute, and or if one of us can unmute you, and maybe you want to take the first question or response, and then I'll get right into calling on some of you who have already put in questions in the chat.
Sure. So how do we do this? Pick the first one here?
Well, Doug, I was just actually wondering if you had any particular questions.
Yeah, you did kind of answer the question of the reduction in all-cause mortality and morbidity is somewhat related. It is other infectious diseases. It's not just all mortality and morbidity. But I was thinking about, I would assume there would be a time component to this, that the further out from the vaccination, the more the effects would diminish. Is there any data to suggest one way or the other?
Yeah, it's a super good question. We are limited often in our studies by the fact that other vaccines are given. So our follow-up time is not as long as we could have hoped for. And one important factor to mention is that once you give a new vaccine, you can alter you can neutralize whatever non-specific effect the former vaccine had. So we really only look at vaccine effects up to the time point where a new vaccine is given. So in most cases, we have a follow-up time of maybe up to 6 months, and it's hard to predict what happens out after these 6 months. But I can say, particularly for the two of the oldest vaccines, the BCG vaccine against tuberculosis and the smallpox vaccine, that there we have seen that having a scar after these vaccinations is associated with survival benefits up to 45 years after you got the vaccine. These are intradermal vaccines that might be very specific for these vaccines, but there we see effects which are also sustained after you receive other vaccines.
Yeah, and I was also going to comment. You mentioned about whether we should stop using these vaccines when the diseases are eradicated, and I think at least in developed nations there might be some data there that could be looked at, because of course I know in the United States, from my own daughter getting vaccinated, I think her first dose of polio was oral, and every subsequent dose has been the inactivated. And so I think now all they give is the inactivated. And so I think we're down to what, measles and chickenpox are the only live ones that are now given. Yeah.
RSV, I think you give also, sorry, rotavirus is also.
Yeah. Okay. Yeah. So we definitely, I mean, that's always, for me, it's always that question with live attenuated vaccines is, the pushback you get is, well, the risk, right? The side effects, the concerns, of adverse events is higher with those. But this offers a potential benefit, which is not something you're going to get with a killed vaccine. Yeah.
Thanks, Doug. Fred, Fox, you were one of the first to chime in with a couple of questions related to interactions between oral polio vaccine and BCG. Would you like to pose your questions?
Sure. I'm a family and public health physician. So in the study where you looked at OPV with or without the early BCG, the BCG provided better nonspecific effects than the OPV alone, but that's also a live attenuated vaccine. So any thoughts on why that occurred, why the OPV alone wasn't providing those same effects?
BCG had an added value to OPV, but thank you for raising the question, because we also did a trial where we gave children the usual OPV at birth, or we didn't give it. We had some indication from a small observational study that actually OPV could be harmful to boys. So we found it justified to do this randomized trial, but it turned out that in the randomized trial, the first observation must have been a chance finding, because in the randomized trial, it came out quite clearly that those receiving OPV plus BCG versus BCG alone had lower mortality. They had, in fact, almost a third lower mortality than those who didn't receive the oral polio vaccine. So this was an important result, I think, in the context that all polio vaccine is soon to be phased out. The plan is to stop it globally in 2024, exactly for the reasons that Doug Reed mentioned, that it's perceived to be a vaccine that can create problems in certain situations, but these very rare vaccine-derived polio cases are a very small number compared with the if these findings are true, the enormous benefit for the babies, non-specific benefits of receiving oral polio vaccine at birth. And we have some of our most recent studies are also on oral polio vaccine campaigns, which have been conducted biannually in Africa for the last decades to eradicate polio. And we can show that these polio vaccine campaigns are associated with mortality reductions of 15 to 25%. So oral polio vaccine is definitely a great vaccine too, with and the latest kid on the block, so to speak. We haven't studied it so much before recently, but it really turns out to be hosting or having some of these beneficial effects too.
Yeah, it seems like that would be a tough sell though,
Thank you.
given the side effects, right? For the, yeah, I mean, it's what, 1 in a million that gets polio?
No, it's less. It's yeah, I can't remember the actual figures. They are in the
Yeah.
science paper, and I know it. I know Gallo and Chumakov tried really hard to get permission to do an all polio vaccine against COVID trial in the US, and they weren't. There wasn't any
It'd be
support
tough.
for reintroducing oral polio vaccine, in spite actually of the fact that if you have received inactivated polio vaccine, it completely removes the risk of getting
Right.
polio-derived disease.
So I started there. Ed LeGrand had a hand raised, and so we'll go to him next, and then I'll give you a heads up that there's basically a constellation of questions related to potential protection against COVID. So we'll go to Ed, and then you can think about those next.
This is great in that what I was paying attention to was the difference between the live vaccines and killed vaccines. And what I've been paying attention to is, why we have, why COVID got out of control. And I think of that as a positive feedback system, just like any infection. It starts out with exponential growth. It starts out very slow, not a big problem. We don't want to put too many resources into it. And then it gets out of control. So all infections, whether within a population or within an individual or fires, if you have no experience with these, they're not worth it doesn't make sense to devote too many resources to them. If you do have experience with them, you realize what a big threat they are. And so COVID came along, we didn't recognize as a positive what a positive threat this would be. I think the next time it comes around, we will. So now our own immune systems have the same problem, figuring out, is this little tiny infection a problem or not? And so it makes sense that, especially in an early childhood, getting experience, an immune system getting experience with an infection that can get out of control would be very helpful. And so that's sort of a one type of explanation for why a killed vaccine is not doing that. It may even be harmful because, like, oh, this foreign material never was a threat. We don't need to devote many resources to it. But if you give a live vaccine, it's like, whoa, this is an exponential growth, and now we need to treat it as such. There was a previous question about, would these effects diminish with time? If I'm right, it certainly would, because I think this is very important early in an individual's life to have some kind of experience with a positive feedback system and exponential growth type of infection. So just throw that out. Thanks.
Thank you very much for the comment, and I think that's an intriguing suggestion, that if you meet something which isn't really dangerous, and in the early child years, or months, that could actually tolerize you. I don't know if it's limited, or I think tolerization is somehow very important, but it might not be limited to the infant period though, because we saw, I just wanted to briefly mention that just as we saw for BCG, that it could train the innate immune system positively, we actually did an experiment with Behind the Tears group where we took Dutch women, and we randomized them to BCG or DTP vaccine, DTAP, or both, and then we did a crossover afterwards. And we could see that in the days after vaccination, actually the first weeks after vaccination, but whether they received BCG vaccine, the live vaccine, or the killed DTP vaccine, they had an innate immune activation. But when we came to the 3-month time point, then those who had received the inactivated Tdap, they had actually tolerance towards other infectious challenges, both innate tolerance and also adaptive tolerance. So I think you're right that tolerance somehow is key, and I'll be happy to think more about it. I like your thought that this could somehow be because they aren't really a threat, and they become, the immune system becomes a little bit negligent about these, interesting thought. Yeah, but tolerance, yes, I think is might be central here.
So I see there are hands raised, but I would like to get to some questions in the chat first. So I'm going to try and tackle a few at once. So could you just comment on what you're thinking about right now about what's known about populations that have varying vaccine exposure to other to other viruses or pathogens and protection against COVID?
Yeah, we had a conference, the first World Conference on Non-Specific Effects of Vaccines in February 2020. And of course, we were 100 people there excited about non-specific effects and talking nonstop about whether we could utilize these effects against COVID. We talked about using them as a here and now treatment, but it was a surprise actually to me that there were so many studies coming out right after suggesting, from groups I didn't even know about, suggesting that childhood vaccination programs, for instance with BCG, could be predicting outcomes of COVID. But I have to say, as much as I like the idea that people were studying non-specific effects and believing in them, I was very skeptical from the beginning about these ecological studies. And I think as time has gone by, we have found out that the data wasn't so strong. It was just because the pandemic kicked off later in Africa and particularly in South America that data looked as if there was an association. I don't think it's there anymore. And so even though I have seen the effect of BCG up to 45 years of age, even for me, it's quite a long stretch to believe that if you were vaccinated in childhood with BCG, that would still affect your risk of dying when you're 80. And basically the countries that do use BCG didn't start when the people who were 80 were young. So there are two hypotheses. It's recent BCG and it's BCG in childhood. And I would definitely put my money on recent BCG rather than BCG in childhood.
Recent BCG rather than in childhood may have an effect. Is what you put your money on. Thank you. I just to switch gears a little bit, another from the earlier from the chat, Dr. Crespi had a really interesting question. You wanna take it?
I was just wondering a little more about the negative effects of the dead vaccines and what could be causing those. I mean, the live vaccines, it makes sense if the immune system is doing something that's clearly adaptive. But there's something unusual going on with the negative effects, which could be because the vaccine itself is novel. I mean, we don't get dead bugs getting injected into our body, as it were. And that also gets to the question of what's going to go on with the mRNA vaccines, which almost seem a little bit more like cancer antigens getting into the body, and should get treated by the body in a rather different sort of way as a result. So I guess there's several questions there, but the main one had to do with the negative effects and then the adaptive significance of the different forms of vaccine with regard to the observed and predicted effects.
I'm not sure I got that exactly. So can you repeat, please?
I'll just repeat that sort of the main part was you saw these negative effects, non-specific effects for the not live vaccines, and they were especially in females.
Yes,
I recall. And is there any sort of known or hypothesized reason for those effects? Because they're odd.
Yeah, I think we came closer seeing that the non-live vaccines, at least in the experimental setting in Holland, could induce tolerance. So if you get repeated doses, I think is also a feature. And actually, we can show that epidemiologically that female versus male mortality increases with each additional dose of DTP. So these repeated doses of non-live vaccines may induce tolerance in the immune system and in a manner so the immune system becomes more lazy when it meets other pathogens, and that will let the bug in and lead to increased mortality. The reasons for the sex differences there are many candidates. There are genetic differences between males and females. There are many of the immune genes are actually situated at the X chromosome. Females have to have a different immune system to become pregnant. Also, pediatricians will acknowledge that females and males have different morbidities. Boys have more respiratory infections, and girls have more autoimmune diseases, and so on. So there are many things that actually allude to the fact that boys and girls might benefit from different vaccinations programs in reality, but we haven't nailed the exact reason why females should be particularly susceptible to negative non-specific effects of non-live vaccines. But the pattern is so consistent that I do think we're onto something, and not least when we recently reanalyzed the data from GSK on their new malaria vaccine, a vaccine that was protective against clinical malaria, but which was actually associated with a tendency for increased overall mortality. And the data was not presented by sex, but we were speculating and wrote in response to the paper, could this be a negative non-specific effect? Because if that was the case, we would anticipate that females had higher mortality, that this lack of effect on mortality was due to negative non-specific effects in females. And it turned out to be the case. So there was a beneficial effect on overall mortality in males, but 2 times higher female mortality among those who received the malaria vaccine versus those who didn't receive it. So the ability of this paradigm to actually predict this event makes me quite sure that we are onto something which is biological. We just don't know the full explanation yet.
I just want to add, that's fascinating. And I think we can probably do our homework to find that article ourselves, but it would be good to add to this chat if someone's got it at their fingers. That's really interesting finding. We have several folks who've been waiting patiently with hands raised. Joachim Kurtz, you want to go now?
Yeah, thank you very much. Fantastic talk. Thank you. You showed maternal priming effects, and you were motivated to search for them by studies in invertebrate and plants. And in insects, there are also paternal immune priming effects. And I was wondering whether you also searched for those and didn't find them, or whether you did not search for paternal effects.
Paper being submitted as we speak, and we have low power because this is Guinea-Bissau. We went back to our old cohorts and reinvestigated parents for both mothers and fathers for their BCG scars, and fathers are not a very loyal group in Guinea-Bissau, so many of them had vanished, or they were at work, I should say. But we didn't have such a big sample size for fathers, but we do find paternal evidence for paternal priming effects, and we're currently investigating it in mice also, where we can do all kind of, vaccinating males and females. And of course, the super interesting question is whether this is something that is transferred via placenta or whether it's epigenetic and could actually also be transferred epigenetically from the father.
Thank you.
You're muted, Born.
Yeah,
I am muted. Molly, Fox, do you have your hands raised?
Oh yeah, are you able to hear me?
Yes.
Oh great, thank you. Thanks for such an interesting talk and discussion. I was just wondering if you could describe a little bit of what the effects on the immune architecture are from these that are associated with these long-term protective non-specific effects, and how that is similar or different maybe to the old friends kind of effect where we're used to seeing people who grow up in geographies that have a diversity of non-pathological microbes, and whether there's whether this is kind of functioning in a similar way.
Yeah, and just I want to amplify my own question, adds really well on that. Is there a specific sort of cytokine profile following a known challenge that you think might be predictive of these non-specific effects?
Yeah, so I mean, I'll just respond to your question first, because of course we have tried to look for immunological correlates of lower mortality and morbidity, and it is not as simple, I'm afraid, that there is just one marker that will give away whatever will happen in terms of your mortality in one cytokine profile or whatever. But what we do have a study where we looked at cytokine profile by 4 weeks of age, and then on mortality up to 12 months. and could see that it was a U shape for most cytokines, really. So you didn't want to be too low or too high, which is actually makes a lot of sense, but it's not doesn't make it a very good marker, I think, in that sense of mortality and morbidity. But what might be a good marker of a well-functioning immune system and also mortality and morbidity, and thank you very much for that question and for making me smile at your pretty little baby. It's a wonderful photo. But that is that we have actually seen some of the same effects of live and non-live vaccine in a Danish context when we looked at atopic dermatitis. So I do think that they reflect some of the same spectrum as, I mean, what we look at in a high infectious disease pressure context in Guinea-Bissau and see in terms of mortality and morbidity might actually be reflected in allergy in high income settings. So I think it's very related hypothesis, this kind of old friends idea, and again bringing me back to maybe we should continue to vaccinate with these pathogens even if we don't meet them on a daily basis. They do seem to carry some important lessons for our immune systems.
Thank you.
That's very interesting. Can I just ask a quick follow-up about that? I just wonder whether it's that the effect, like maybe it's not specifically about the pathogen in the vaccine, but the fact that the vaccine is evoking such a low grade immune response, and it's just that response that educates the immune system. Is that possible?
I would say both with BCG and smallpox vaccine, you do have some sustained infection for, I mean, I'm personally having my third BCG. I had that recently for like a month ago or so. I'm a believer in my own theories. So, and there I have this wound that continues to leak and I know it will do that for maybe 6 months. So there is some sustained low grade activity, but I'm not sure that would be the same for measles, for instance. I'll have to think about it because normally people wouldn't anticipate that would stick around for very long if you give a measles vaccine. But worth thinking about. But yeah, and definitely we have shown quite recently that BCG enters the bone marrow and might also stay around even longer than you just have it in that ongoing skin reaction that you have for some months. It might enter the bone marrow and train your progenitor cells there.
I think with the increased tools that we have these days, we're starting to find things we didn't expect. I was thinking of, well, if you look at the Ebola outbreak in West Africa, you know, there had never been any thought that Ebola could be a latent disease. And yet now we have evidence that Ebola can, in fact, survive in the vitreous humor of the eye and reemerge. And in my own lab was some work we've done here that we just published one paper on the Eastern equine encephalitis virus. And we have a companion paper on the Venezuelan equine encephalitis, in which macaques that survived infection out to 28 days, and in case of Venezuelan, out to 56 days, we can still find evidence for the virus in the central nervous system, even though there's no indication of any disease in the animal. So these viruses, even though we call them, you know, acute, may persist a lot longer than we realize.
Fascinating, yeah.
Thanks, Doug.
Yeah.
Bettina, do you want to, hopefully we have time for one question after this, but this might be it. Let's thank you everybody and those who've been waiting patiently. I'm sorry I haven't gotten to your questions.
So hi, and thanks for a great talk. I'm a total non-expert, but I have the following question. Are there any data that compare non-specific effects from natural infection to those of vaccination?
Yeah, and actually I was thinking of that as a follow-up to the last question also, that the infections themselves probably also carry these non-specific effects. So we have shown for measles infection that once you survive the acute phase, you actually have lower mortality than if you hadn't had measles. This has been shown in 5 studies and in all the available data from the original period where you had a true comparison of naturally infected versus non-vaccinated or non-vaccinated and non-infected, then there have been some recent studies saying that natural infection actually has long-term negative effects but they are comparing versus being vaccinated. And that's a different kind of assessment. So really going back to the old days where you could compare those who were naturally infected with measles with those who weren't either vaccinated or had measles, there you saw non-specific benefits of having the infection. So I usually say to people when they're skeptical about, and that's funnily enough, it's MMR vaccine that most people are skeptical about. I say this is actually your great chance of having these natural infections in a mild way where you're guaranteed to pass the infection without having any side effects or very little risk of side effects, but you get the same benefits as people usually, and I think rightly perceive as the benefits of passing the natural infection. But of course, the natural infection comes with this risk, which is absolutely not small in many settings of getting really ill from measles.
Well, I hope that partly addresses your question, Ronald, who are waiting patiently about thinking about effects of natural infections. And
one thing I could maybe just throw in is that I'm curious about whether that's one speculation I have, because most studies that have looked at the effect of treating latent tuberculosis actually, unless you're a child or immunodeficient, suggest that it's harmful to change or can be harmful to treat latent TB. So latent tuberculosis infection, if it doesn't make you ill, it actually might look as if it makes you stronger. So just along the same thoughts that it can be beneficial also to harbor the pathogen if it doesn't kill you.
Well, we've reached 1 o'clock and there are still more than 100 people tuned in here, and the comments are still coming in. Christine, this is a tremendously exciting and provoking presentation. We're really grateful. So on behalf of all the centers and everybody else in the TRISEM community, thank you. Meredith, do you want to end with any final remarks about what's coming?
Yeah, I'll turn it over to Charlie. He probably has the website up and ready to show.
Sure thing. And I'll just say, Christine, that was a fantastic talk. Thank you so much. Vaughn, thank you very much for moderating, and thank you all for all the great conversation and questions. This was really a great success, so thank you very much.
And I'll just throw in that if there are any questions that you didn't get to ask me, you're very welcome to write to me, and I'll be happy to answer them by email.
Excellent.
And thank you so much for the interview.
And I'll just share quickly our website. Hopefully you're seeing that now. Here's the talk we just heard. Fantastic talk. And next week, we think we have Phil Starks on tap to speak on the 17th, Wednesday at 1 o'clock. We don't yet have a title there, but we're optimistic that we'll be hearing from Phil. After that, we're going to be hearing from Robert Woods from University of Michigan. Does evolutionary biology have anything to say about how we should use antibiotics? That should be interesting. You can scroll down and see the other talks that are coming up. We have a great lineup, including my colleague Herman Ponser, right after we hear from Bob Woods. So please, join our listserv if you're not already on it, and stay tuned for the next talks. We hope to see you at our next Club EvMed event. Thanks everybody for tuning in.